and thank you. I am very pleased to be here. I'd like to thank the organizers for letting me share this recent work that's gone on for a few years, actually, and we're finally getting this all together into stuff that other people can use in reporting on it. So this has really come out of work with the both my PI's lab, Chad Myers, and our collaborations with Charlie Boone at the University of Toronto, and also collaborators at the Riken Institute in Japan. So just to give you a brief overview of where I'm going with this talk, first I will provide a brief motivation for why these computational tools were designed and implemented, and then from there I will describe the two tools that I'm presenting to you today and give a short wrap-up. The basic motivation for designing the software comes from these chemical genomic screens that we have, that our collaborators have performed, and we've been primarily involved with the analysis of the data. And the questions that we are asking are very basic. And basically they're what happens when chemicals interact with biological systems. People have been asking these questions for a long time. We have a complementary experimental approach to try to get at, to ask these questions get at new, potentially insightful answers. And the system that we use is a system involving the yeast genome deletion collection. And as you can see here, each one of these budding cells represents a yeast with one particular gene knocked out, different gene for different color. The type of data that we work with are these profiles in which the red indicates that a particular knockout strain is sensitive to a particular compound when we put the compound on those cells. So these are, you'll see these as chemical genetic interaction profiles throughout this presentation. And what this really comes down to is it is a high-dimensional functional profile of what a compound does to a cell. Depending on what the compound does, hopefully, and we do see that different strains will drop out if the mechanism of the compound is different in those cells. Now, it's not just the fact that we had some profiles that necessitated writing the software. We actually had generated a lot of data. So just to show you the type of experiments that we run and how we generate the data, we take this barcoded deletion collection that I showed you the individual strains. We put all those strains together into a pool. We put 96 pools in a plate, and in each different well we put one chemical on there. We isolate the genomic DNA and multiplex these together via PCR, so that at the end we get, in a sample, we get a bunch of different PCR products with different barcodes and different index tags. The barcodes map back to the strain, and the index tags map back to which condition that PCR product came from. And I'll stop right here and say that the software, it was actually, I'll reiterate this, it's generalizable to anything that works on, for any data set where this type of PCR product is involved. So we designed it for this type of experiment, but I'm sure there are other people who are working with barcoded collections of some sort and multiplexing these things together. If your PCR product looks like this, you certainly may like using this particular pipeline. But this particular multiplexing step allows us to put 768 conditions together into one high-seq sequencing lane, and that's how we generate this large amount of data. And we end up at the end, after doing the sequencing and data processing, with these pipelines. So what is the processing that leads these pipelines? That is the first piece of software, which is we have called BeanCounter. And to show you a general overview of what this pipeline is, it takes in the FASTQ files from the sequencing, and it also takes in, most importantly, mapping of what the index tag to, a mapping from the index tag to condition, and a mapping from the barcode from the knockout strains to what that strain actually is. And we put all these things together, and the software starts by parsing the FASTQ files and counting the index tags and the barcodes, and putting together a matrix showing which strains had a certain number of counts in each particular condition. And then we score the interactions, which I'll get to in a second, end up with z-scores, and then we have a modular post-processing pipeline that allows for supervised correction of batch effects, such as index tag and lane effects. So correlations that when conditions are have the same index tag or in the same lane, they're a little too correlated than they should be. Otherwise, we have, you're able to correct for some effects that are not explainable by technical or other biological effects, but you might still want to get rid of. and we have ways to evaluate all those corrections and visualize the data at every step. To give an example of what the raw data look like when they come in, this is a profile for mycofungin, which is a compound that perturbs the building of the yeast cell wall. What we see is, what we do is we take the experimental controls and we calculate a mean profile, and we compare each compound condition to that mean profile. And then you can see here, these are particular notable dropout strains. BCK1 is by far, at the end, you will see really actually drops out the most. But what we do is we do a couple of things. There's a lowest normalization that goes through the middle of this to provide a normalization. And then we estimate the variance, we calculate a variance of just based on the condition itself, and also a variance that depends on all of the DMSO controls, and it depends, it's a function of where we are in the read count. So in people doing RNA-seq stuff, we know that variance as a function of read counts is an issue. We definitely take that into account, so we get a continuous estimate of variance as a function of the read counts. And at the end, we get chemical genetic interaction z-scores that we, these are just plotted against the mean profile here again, but these are the z-scores that we then take into further steps of the pipeline. After we calculate these z-scores for one particular data set that we have here with 1,000 different chemical conditions and 3,000 yeast strains, these are what the data look like. And some of those notable interactions, it's kind of hard to read, but they definitely show up as the red here, which is a negative chemical genetic interaction. A couple of the effects that I want to show you this pipeline is capable of removing are, one of them is this concept of batch effects, which I mentioned before. In this particular situation, again, it's hard to read here, but all of these conditions actually have the same index tag. So there's something going on here. In this particular condition, we notice that there actually seems to be an interaction between the identity of the index tag and the identity of, or the first base of the particular barcode. That's not always the case. There are definitely other ways to see batch effects in these data, and our pipeline provides ways to take those out, as long as you are able to encode in the table of information about all the samples what particular batch or lane index tag that that condition maps to. After we kind of correct for the index tag effects and other effects that may be there, we have, you may notice there's a large source of variation in this data set, and we actually have not been able to explain it biologically or technically, and just decided to implement a way to remove it. And since it is the largest source of variation in the data set, it's very easy to remove via singular value decomposition, just taking out the first component of variation. And throughout this pipeline, I'm going to show you a snapshot of ways that we evaluate all these corrections. This is an ROC analysis. What you can see is this is an ROC of compound-compound correlations. So when we what you would want is, if you have multiple biological replicates of compounds in the experiment, you would expect them to correlate. And actually what we see here is, before removing this first component, the correlations do not look that great. Having a high correlation is not the most predictive of being treated with the same compound. Once we remove that, we see that the largest sources of variation was actually kind of masking those correlations. So in a lot of our data sets, we remove one component right off the top to get these correlations looking much nicer. And as you can see more qualitatively too, I think this data set looks much cleaner, and certainly we know from other things that we are still retaining information in this matrix. So, and just to reiterate all those, well, I'll just go through the summary here. So what we've got with BeanCounter is we have a pipeline to process any type of barcoded, multiplex barcode sequencing data, inspired by our chemical genomics analysis, but I want to emphasize that it is generalizable. The interaction scoring incorporates variance models based on the variance of the strains that counts in the condition, and also variance as a function of the read count abundance. And all the steps at the end for post-processing are modular. You can put them in any order and visualize and evaluate the effects that those corrections have at every step. Now that we have a way to generate these chemical genetic interaction profiles, we wanted a way to interpret them in a biological biologically meaningful way. The core idea of this pipeline that we've dubbed CG Target is that chemical genetic interaction profiles should be interpretable using genetic interaction profiles. And since we're working in yeast, Saccharomyces cerevisiae, we have the largest genetic interaction network that we know is very informative to date. The Myers lab has been involved in scoring those interactions for quite some time now. But the basic premise is if a drug or a compound's chemical genetic interaction profile is similar to a genetic interaction profile, it looks like the drug is knocking out this gene. These two profiles are exactly the same, except here you're treating with the compound, and here you're deleting a gene. So compound phenocopies the gene knockout. To implement this, the pipeline has a few kind of basic steps. It takes in the chemical genetic interactions. They could be from BeanCounter, they could be from a different source. The genetic interactions, in our case, from the yeast genetic interaction network, and a mapping from gene to go biological processes. First step is to compute correlations between the chemical genetic interaction profiles and the genetic interaction profiles. And then from there, we score and evaluate the significance of these correlations when we look at how those correlations map to the go biological processes. And finally, there is a step to estimate our false discovery rate. To give a little bit more detail about this process, first here's the step where we perform the correlations. We map those correlation scores onto a particular gene ontology biological process term, and at the end, we basically get a matrix out that maps the particular compounds to the biological processes that we believe they perturb. Since there are many computational people here, I'd like to go in a little bit more detail about how this works. And it's really important when we were developing this because what we do first is we take control chemical genetic interaction profiles, and we actually use 3 different versions of control genetic interaction profiles. First are the experimental controls, so conditions where we just treat with the solvent, we don't treat with the compound. And in one of our large screens, for example, we have like 5,000 of these, so there's a lot to work with. But then we also generate profiles by resampling the entire data set of the conditions where we actually treated with the compounds. So we take to create a new profile, we take one value from each strain chosen randomly across all the conditions. And that kind of accounts for variation in the strains that we see when we actually treat with a compound. And then we also shuffle the gene labels on each individual chemical genetic interaction profile to give us an estimate of what happens what variation is introduced when we just have a profile that has those particular values in it. So we use all those null models to make sure that in no case could we randomly get the particular scores that we're seeing in our analysis. So once we make these distributions to compare against, then we compute similarities with the actual compounds of interest. In this case, Benamil, which is a tubulin polymerization inhibitor, we see that its score is way outside the distribution. So this is a very, very strong prediction. It actually beats every single randomization, all attempts to randomize that we put at it. So once we do this for all compounds across all particular go processes, then we take the resample profiles and the experimental controls and kind of run them back through the pipeline and see how many times we get a certain number of predictions at a certain level of significance. So in this case, these are the actual number of predictions we get for the compounds. This is the number of compounds that we get at a certain p-value. And what you see here is we now have a way to kind of continuously draw a cutoff at a certain level of false discovery. So in this case, you will see that at about here, that looks like about a 25% false discovery rate, and we get about 850 conditions out of this particular process prediction run. And what you'll also notice is that using the resample profiles is a much more conservative estimate of the false discovery rate. If we were to use only the experimental controls, which a lot of us would agree are in many cases are totally valid to use as controls, we would say that almost all of our predictions were actually of a high confidence nature, and that we were definitely suspicious of that. There are definitely a lot of other effects that happen on these cells when we put compounds on them. So certainly using this this method is good to provide a realistic estimate of the false discovery rate. Now to actually ask if we are making good predictions, and the answer is definitely a yes. So what you can hopefully see in this table is for each of these compounds, the top ranked prediction is something that is something that definitely nails what we know the compound to do. So for example, latrunculin B is an actin inhibitor, and that definitely should inhibit the movement of cellular components around the cell. So we go down this list. There's only really 2 examples of things that don't work. And again, sometimes those are actually limitations of the experimental method, not necessarily the process prediction method. We have applied both BeanCounter and CG Target to a large number of chemical genetic interaction screens. These are the diverse collections that we've screened. And in total, more than 53,000 samples have been analyzed. We have computed more than 15 million compound mutant interaction scores. And at this point, more than 1,700 compounds with high confidence predictions have been identified. Once we get these predictions, I just wanted to show quickly, we have these functional neighborhoods of the yeast cell. And it is really interesting because all these different diverse collections, we definitely see that, these collections don't just hit the compounds in those collections don't just hit one particular part of the cell. So they have these diverse predictions, but also you can see that their signatures are rather unique. And we have more quantitative ways to address that I've talked about different places. But certainly we have diversity, yet each of the compounds in these collections we can tell that they're specific for certain functions. So to conclude, both of these software pipelines we have written scalable pipelines to both process the raw data and to predict the process level targets. It has been an enabling technology for us, and we hope for others to perform large-scale barcode sequencing, discover novel compound function, and both at a single compound and at a library level, characterize the function of compounds. And in the future, we will be applying this to different types of yeast collections and also to screens in different species, as well as I'll be certainly looking at the processes of CG Target to see if we can use different types of gene sets to get target predictions, as well as what the accessible target space is. Do we have blind spots in the predictions that we make? So with that, I'd like to acknowledge all of the collaborators and people in my lab, major people in this work: Chad, Justin and Hamid from our lab, Sheena from Toronto and Rican, Jeff, who's moved on to a startup pharmaceutical company, and certainly the funding sources, especially the MPGI at the U of M for a generous travel fellowship. So thank you.